FDP, Netherlands, and industries. He is actively involved in the field of drug delivery, which includes improvement in oral bioavailability of BCS class 2 and 4 drugs by polymeric conjugates and nanolipid based system, nanoparticulate drug delivery system for brain delivery for the treatment of CNS disorders, nanoparticulate system for phytoconstituents. He is supervising scientific research at the post-graduation and the doctoral level. He has guided 76 MPharm and 37 PhDs. Presently, 2 PhD theses are under his supervision. He is an external expert for PhD theses of a large number of Indian and foreign universities. He has written several books and has contributed several invited book chapters on controlled and novel drug delivery systems in edited books of reputed international publishers. A widely traveled person, he has presented his research work in more than 100 conferences held in India and abroad. He has produced over 350 manuscripts in journals of repute and 5 Indian patents granted applied. He has an H index of 68, ITAN index of 245, and more than 17,800 citations to his credit as per Google Scholar bibliometric data in August 2021. He is on the editorial board of many renowned journals. He has guest edited couples of thematic issues of reputed international journals. He is a reviewer to a large number of leading international and national journals of repute and book proposals from various international publishers. He is an expert for project reviews for agencies from India and abroad. He has been featured in recently published worldwide ranking of top 2% scientists, pharmacy and pharmacology category, also named in the top scientists in global list across all fields based on an independent study done on published by Stanford University in 2019. He has also worked as a paper coordinator of UGC e-Pathshala project of the Ministry of Education, Government of India for 2 papers of pharmaceutical sciences. Professor Ali is also actively engaged in committees of various universities, councils, accreditation bodies, public service commissions. He has been an executive member of Indian Pharmaceutical Association Delhi branch from 2012 to date. He has been a member of local organizing scientific committee of conferences. He is a and has organized staff development, refresher, short-term training program from time to time. He is a life member of 20 Indian and international scientific organizations. We are honored and privileged to have you with us, sir. I welcome you, sir.
So thank you, and I think now good morning to all of you. Today it is raining heavily. I think because of that, there may be certain net issues. So at the outset, I would like to thank Dr. Praveen Kumar Gaur Sahab, the Director ITS, for inviting me and interacting with you all. Thank you, Sagarika, for giving an elaborative CV of mine. I thought you were going to shorten it, but you read all the contents over there.
Sir, it's so nice. I can't miss it out.
Thank you. And since the theme of the ATAL FDP is based on drug engineering, so I thought that I should talk about certain nano carriers which are being developed in our lab as well, right? Starting from the challenges and the opportunities which we have explored through the funded research and so on. But generally, whenever I'm going to FDPs, I see that there's a mixed population of fellow faculty members who belong to various groups. So it becomes very difficult because many a times, the area in which we are talking about may be not of interest. But I think this is applicable everywhere in all branches, and this can be explored in that way even, right? So I'm just sharing my screen. Just give me a moment and then I will start, okay? Can you see on your screens the first PPT?
Not yet.
I can see on my screen, and it
Sir,
is coming. You
it's
are
not
screen
yet
sharing.
visible. Yes, sir,
Not
it's
visible.
visible now.
It's visible
So
now.
visible, visible.
You will find certain
अच्छा
lag
नहीं है,
because of the net
सी तो
connectivity,
अच्छा
because
है
of the
जो
weather.
पहले आया था.
I will request all participants to mute themselves, right? They can mute themselves, and then if there are any queries, they can write in the chat box, and accordingly they can be taken. Otherwise, this will break the link as well, right? So anyways,
Sure,
now
sir.
I'm going to talk about lipid nanocarrier for bioavailability enhancement. What are the challenges and opportunities which are available? This is a piece of work from one of the ICMR funded project, and how we initiated and how we started. This is being presented over here. I think today there is a problem. It is yeah. We are going to we are going to talk about the lipid-based drug delivery, how it originated, what are the immediate limitations, and what are the strategies which are available, and what are the advantages from the point of view of the drug delivery systems, right? What is the unique structure of NLC because of which the scientists are taking interest in converting into a drug delivery system? What is fate after oral administration, and what are the methods of preparation? Very briefly, I'm going to touch upon, and some experience in our lab, optimization and characterization of NLC and in vitro and in vivo study, as mentioned already, that it is an outcome of an ICMR project. When we talk about lipid-based drug delivery system, we, as a pharmacist, started with the very coarse type of lipid-based drug delivery system in form of emulsions, which are, I think, known to everybody. Two immiscible, one lipid and one aqueous phase mixed together with the help of an emulsifier, forming a liquid emulsions. And this is one of the good type of a formulation which was taken by patients in the form of liquid dosage form. And then after liquid emulsions, there were a lot of transformation. Liquid emulsion changed into multiple emulsions, right? And then multiple emulsions going into micro emulsions, and then nano emulsions, micellar systems, and so on. And then the major second generation lipid based drug delivery system which evolved was solid lipid nanoparticles, which was based on solid lipid matrices. As the name indicates, it was having only a solid lipid which was of maybe of physiological origin. And in order to obviate what are the challenges we are going to see with the SLN, what were the problem with the SLN, and accordingly the evolution came for coming up with nanostructured lipid carrier, which includes a binary lipid. One of the component is the solid lipid, and the another one is a liquid lipid. The reason for incorporation of a liquid lipid was that it is going to create imperfections, and those imperfections may lead to a high drug loading, which was not possible in case of solid lipid nanoparticles, right? So this irregularity in the structure was a beneficial step toward for the scientists who were able to have a greater drug loading, as because of the infections or imperfections also, there was an advantage of stability issues also. We were able to get a higher physical stability as compared to solid lipid nanoparticles. Liquid emulsions, they consist of liquid lipid matrices, you already know, and then they include a high level of surfactant concentrations, and then they are physically unstable, and on long-term storage could lead to oswald ripening. And then you have studied about formation of agglomerates, and then floccules, and all those issues, where the particles agglomerate and the particle size increases and so on. So after this, there was an origin for solid lipid nanoparticle, which was composed of solid lipid matrices. They were having limited drug loading capacity because they were having only one solid lipid crystal structure, right? And this was a perfect structure which has no scope of accommodating a higher concentration of drug levels, right? And over storage, what happens that there is a drug expulsion during storage because it's a perfect structure, and perfect structure generally try to remain in the state of homogeneity, and in order to remain in the state of homogeneity, they will make the drug to expel. And then comes the latest, series. Maybe, in the coming sometime, you will find certain other advances coming in the nanolipid carrier. Is a nanostructured lipid carrier. As mentioned already, it is a binary lipid, and it has an advantage of improved drug incorporation and the release properties. In addition to it, you can modulate the release profile, and you can also have a higher stability, which was not possible with the SLN preparations and the other lipid carriers, maybe the conventional ones. The issues of concern with the conventional lipid-based formulation was poor drug loading capacity. You are using high levels of excipients, surfactants, co-solvent, co-surfactant. There was a problem of precipitation of drug, upon aqueous dilution in vivo. When the formulation was taken, there was a problem that when it comes into contact with the higher volume of the upper GIT, that is the gastric fluid, the drug precipitate, and it is difficult to get your drug absorbed if the drug is in the precipitate form, because it should be in the solubilized form. There will be expulsion of drug during storage. In the container there will maybe expulsion, and then you are using a formulation which is D drug. There is no drug over there because drug has expelled out, maybe within the container, or it is on the surface or whatever. And due to the toxic effect of the surfactant and the co-solvent at high doses, the per day and the per dose uptake level is very restricted, because the incorporation of surfactant and co-solvent is based on the GRAS list. The GRAS list gives a limit as per the route of administration, and accordingly their usage is restricted. I have already talked that the limitations are that they are having a lipid crystals which are not going to make the drug at a higher concentration being accommodated in the structured lipid. However, there was also a problem of expulsion of drug during storage. NLC, a novel approach of targeting drugs, came into existence, and it was reported by Mueller in 2002 and 2007, and some work was also going on earlier also. This is a unique blend of lipids. NLC are new type of lipid nanoparticle, constitutes of a mixture of solid lipid, as I already mentioned, with a incompatible liquid lipid. Incompatible does not mean that is chemically or physically incompatible. That means they maintain their own physical characteristic. One is solid and another one is liquid. Unstable matrix of NLC for advantage of improved drug loading capacity. So this instability is not talking about the stability of the carrier system. It is talking about the stability of the solid lipid and the liquid lipid, which will cause improvement in the drug loading capacity based on the imperfections which are caused there. That you can modulate the release properties based on your requirement, whether you want to have a controlled release or only a sustained release, and so on. There may be in the old SLN perfect crystal, you were having a brick-like structure, which is, which is not, allowing anything to enter into. But however, when you are using imperfect crystals, the spaces in between will make the drug to accommodate them in themselves, right? So that is the advantage because of which we are having a higher stability and higher drug loading. Solid lipid, they fall from the various classes. The basic one is a glyceryl monostearate, and there are so many, brands which are available: Computerol, Peristrol, stearic acid, Cutinia, Gelucire, Anapafil, and so on. Liquid lipid may vary from the long and medium chain fatty acids, right? And then polar oils such as oleic acid, vegetable oils such as soybean oil, castor oil, and nowadays we see a lot of liquid lipids which have certain therapeutic values as well. Maybe vitamin E, which has an antioxidant properties, oil like Nigella oil, which has so many anti-inflammatory and neuroprotective activity, and almond oil and many oils. They are being taken as a liquid lipid, provided the drug has a good solubility in them, and accordingly this will add to the value to your drug delivery system, because there will be a additional advantage using a oil with a therapeutic importance. Surfactants which are being used as polymers, twin 20, twin 80, polyethylene glycol, polyethylene oxide. Now here I would like to emphasize on a concept which is being termed as peak glycoprotein efflux transporters, where when the drug is being absorbed, these transporters are going to efflux the drug from the systemic circulation back into the intestinal lumen. And that's why they are being termed as efflux transporters. So as a scientist, it is, we generally try to find out if the drug is a P-glycoprotein substrate. So in addition to finding out the properties of E-glycoprotein inhibitor in the solid lipid or a liquid lipid, we generally try to find out a surfactant with that property. So many hydrophilic surfactant like tweens, they have been reported to be, to be not identified. They are not being identified with a substrate for efflux, right? So you will find many nanoparticle approaches where these are being surface coated in order to so that they are not being identified and there is no efflux, so that you can have a higher bioavailability. Similarly, you can also do polyethylene glycol coating, which is also a hydrophilic coating. This will also lead to in surface modulation, where you can have the long circulating colloidal particle, which is happening in case of the liposomal preparations also, where the RES system is not able to identify it. It prevents opsonization, and hence you can have long circulating colloidal vesicles. So these are the approaches which are also applicable here in NLC formulations. Also, this is the image which I have already talked about, that there are no imperfection in SLN. So you can see that only a small amount of drug is getting incorporated, and after, just a moment, yes. So after, after getting incorporated, they are expelled as well. But in the case of NLC formulation, these imperfections are leading to drug to enter into and accommodate, place itself into the imperfection, which will minimize the drug expulsion as such. So coming to the advantages, why we intend to go for NLC formulation: high drug loading capacity, improved drug release profile, and stable drug incorporation during storage. So these 3 points I've already taken into, but now I will like to emphasize on lymphatic uptake here. So many drugs candidates who have the problem of first pass metabolism, they can also be the candidate for NLC to be taken by oral route. The reason being, when the NLC comes into contact with the the GIT and comes down, so what happens is that is being treated as a fatty meal. A very best example of busulfan and the fatty meal concomitant administration. And similarly here also, this will lead to the release of bile, right? Bile, and then bile salts, they emulsify these nanostructure lipid carrier. It is being taken by in the intestinal lumen, right? Formation of chylomicron with the release with the lipoproteins, and so the size increases. So what happens is these chylomicrons, right, being in the range of slightly higher particle size, they will not enter into the portal circulation. So this is going to save our drug from pre-systemic metabolism, or we can say that it is not exposed to first pass metabolism, or we can say that it is not exposed to cytochrome P450 mixed oxidase reductase system as such, right? So for those drugs which have first pass metabolism issues, right, we can also give in the form of lipidic nanocarrier, which will be taken by lymphatic uptake, which is interconnected with the systemic network as well. So you can also work with a low dose. So particularly mentioning over here as well is that, for the oral dose and the conventional formulation, you can also decrease our dose to a great extent, which will also decrease the peripheral side effects. So everything is interconnected. You decrease the dose, you are able to achieve the lesser toxicity or lesser toxic effects in cases where the drug have associated risk issues. So the application of NLCs are enormous. They have been tried in almost all. Ocular will have C. So the all type of systems as such, right? And in case of parental application, biocompatibility is important, right? And then protection of drug from enzymatic degradation, but since it is being accommodated inside, so that is not a problem with NLC formulations, right? In the case of topical applications, you will find a lot of topical preparation in the German market, in Germany. skin residence, small size control release, and biocompatibility, facilitating skin penetration of drug and offering occlusive effect and improved skin targeting. So it is basically talking about topical application. It is not talking about the transdermal format, type of delivery where there are so many skin diseases like skin carcinoma, psoriasis, vitiligo, other type of antiviral infections, viral infections and fungal infections. These can also be tried using NLC for topical application because they offer occlusive effect and targeting of skin is good. And NLC for intranasal application because when they are being administered, you can avoid the PBP, so you can rapidly take it, get it entered through the intranasal from palate, olfactory shunt, right? It is a good bioacceptability. As mentioned, they are biodegradable, minimal toxicity, and absence of burst effect also. NLC for ocular delivery has also been tried, improve interaction with ocular mucosa and prolong retention time in eyes as such. Oral delivery, a very well accepted route of administration and having a highest patient compliance, right? So they have been tried, and our whole, discussion in the future slides is based on oral drug delivery. So very quickly, I wanted to give an overview that these systems have been investigated in almost all route of administration, and there are a lot of reports available. If you go to the PubMed search, right, and you put nanostructure lipid carrier, and you can find out how many hits are available, right? Oral drug delivery, when we talk of, this is the theme where we talk about the problems which are associated with the conventional oral drug delivery and what are the steps which can be taken. The barriers for oral administration, the first one, I think all our UG students and our fellow teachers who are teaching biopharm and pharmacokinetics must be aware that these are unstirred water layer lining the enterocytes. So this unstirred water layer will lead to the bulk saturation. So CSS is achieved very quickly, and then the dissolution is a problem. Dissolution, the formulation of dissolution of the particle is a problem. So this is a problem in case of drugs which are having low solubility and a high log P value. The second is a P-glycoprotein mediated efflux. I've already talked about that, when it is taken intracellularly, then also the drug levels are thrown out through this efflux pump. And intestinal metabolism also occurring because of the microflora present over there and because of the enzymatic system present in the intestine. So this is another barrier which is leading to a low bioavailability. So this can also be overcome because you are encapsulating, you are adjusting your drug within the lipid nanocarrier. So this is also going to inhibit or is not going to make the drug exposed to the enzymatic system for degradation, right? And in addition, the carrier which is also there, they are also having the inhibitory effects on this intestinal flora. Hepatic first pass metabolism, you already know, and I have already mentioned that this is leading to the systemic metabolism because of the presence of the enzymatic systems over there. Coming to oral delivery, why there's an opportunity with oral delivery? Because we are talking about physiological and biodegradable lipids, right? And they are also biocompatible. This biodegradability is of importance when we are giving parenteral preparation, but in here, it's okay, this is the advantages which is there with the lipids. They have efficient adherence on the gut wall, prolonging residence time for absorption of drug. They have certain bioadhesive properties, so, you will find a good adherence. There is a reduced dosing frequency because it is due to sustained drug release profile. If the sustained drug release profile is a prolongation, it is sustained release. Otherwise, we can also achieve control release if the order is 0-1. Minimize toxic effect on non-targeted site because you are targeting to one site, and moreover, if you are able to achieve a good bioavailability, then accordingly you can also minimize the drug level. Dose reduction is possible due to their high drug payload capacity, and accordingly associated peripheral side effect can also be minimized. And they are viable commercial potential because of its ease of scalability, and they can be administered in a soft shell, and they can be given commercially as well. Now, what is the fate when I have talked about the formation of chylomicrons and then they are taken into lymphatic systems and so on? When NLC lipids and the bile salts, they mix together, they form micelles. There is a high concentration gradient for efficient diffusion across unstirred water layer. So we are overcoming one of the barrier, right? Small particle size, improved surface area, improved drug dissolution. Then there is a high dispersibility and effective adherence on the gut wall, and there's a prolongation of intestinal residence time. So, and this will lead to a better absorption. Unique anatomy and physiology of the lymphatic plus drug incorporated NLC, direct transport of drug through intestinal lymphatic into systemic circulation. So the important part over here is, since we are using NLC for as a drug delivery carrier, accordingly, the drug inherent property should also be inclined to more lipophilicity. So the drugs which are having a high log P value above 4 or something, right? So they are the best candidates suited for the oral delivery by NLC for lymphatic uptake. This is this I've already talked about. You have the formation of micelles which are being acted upon by pancreatic lipase, right? And there is a free fatty acid monoglyceride, and this combines with lipoproteins to form chylomicrons, which are taken formation of chylomicrons, and these are taken into the lymphatic vessels as such. Drug candidate for NLC, mentioned already, poorly water soluble, highly lipophilic, log P more than 4, and potential substrate for P glycoprotein. So you can always incorporate a P glycoprotein inhibitor, and then they can be extensively metabolized in the intestinal gut and liver by the cytochrome P3A4, which is present in the intestinal gut, and then the drug which are suitable candidate for lymphatic transport. Many times we require certain drug candidates into the lymph nodes and so on, anti-cancer drug. So this can also be an added advantage. And drugs which are quickly eliminated, or the drugs which are having short half-life, we can have a sustained action or a controlled action for those drug candidates. So 5 criteria. So you can always choose a drug based on these 5 criteria and accordingly go for nanocarrier formulation. Method of preparation involve high pressure homogenization, where a drug is dissolved in the liquid melt, which is dispersed in surfactant solution, and the coarse pre-emulsion is passed through HPH at pressure of 100 to 1500 bars. Extremely narrow particle size range is obtained, and the higher lipid particle content in the dispersions also, and you are avoiding any organic solvent. So that is another advantage, right? And scale up is feasible, and but the problem is there is a generation of high heat which may lead to degradation, right? So that is a major disadvantage, but in some of the modification that has also been overcome. The another one is the oil in water microemulsion. You have to take an organic phase in which the drug and the lipids are dissolved. If the drug is, because we are taking NLC, the drug will be always water insoluble, having a log P value high, so it will go in the organic phase only. Both phases are mixed together with mechanical agitation, and NLC suspension is formed. Dispersion of warm oil in water microemulsion in a cold aqueous medium under mechanical stirring. There is a avoidance of high pressure and temperature, and avoidance of toxic solvents. High level water content is difficult to remove. That is a major problem. So always you will have a water content retention, and there is a high concentration of surfactant requirement also. So maybe it will be of importance where we are trying to get something for topical preparation or those areas. The another one is a solvent emulsification, evaporation, or diffusion technique. So you melt the lipids, right? Which is a binary system. How they are being done, that we will see. Is dissolved in an organic solvent, which is emulsified in a water bath under mechanical stirring in an aqueous phase containing surfactant. So the moment it is being mixed up, so there will be formation of emulsion, solvent is evaporated, and stable NLC dispersion is formed due to precipitation of the lipid in the aqueous medium. Advantage is you can get a very small size of particles, right? The range will be, not very high, and you can have a good PDI, polydispersity index, as well as good particle size. And there's avoidance of heat during preparations, and hence drugs which are thermolabile also can also be tried for this approach of drug formulation. Toxicity issue with solvent residue. of the product obtained by using this technique, and then the particle concentration obtained using the technique is very low. The another one is a high shear homogenization. You can use a high shear homogenization rod also, or ultrasonication. So melted lipid is dispersed in an aqueous surfactant solution under high shear homogenization with rotor stator homogenizer or ultrasonication using probe. The obtained dispersion is then cooled down to room temperature. There will be avoidance of toxic solvents and low dispersion quality with presence of microparticles. There may be a physical instability during storage and low lipid concentration. Surfactant concentration is high, and contamination with metals can also take place. So though it is a method, but it has a lot of shortcomings. So it is not very much being tried. You will not find much papers on high shear homogenization or ultrasonication. We have seen, the NLC formulations. They have been tried on number of etoposite, right? The candidate, sorry, and one of them is the etoposite, which is a substrate for P-glycoprotein, and different combination of liquid lipid and solid lipid is used. And they have been tried with the improvement of relative bioavailability. And the improvement is not 1 or 2 fold, it is quite high, maybe 3 times or more than 3 times, right? And vinpocetine, simvastatin, right? Drug like lovastatin, antiretroviral drugs, they have also been investigated, and then tamoxifen, very wide, reporting is there in the literature search engines, right? And these types of the candidates have been investigated, and then they have been tried. You will find a lot of patent search hits also when you go for nanostructured lipid carriers. Some of the products are also available, and accordingly, this is being done. Now I'm coming to, and this is also incorporate the characterization which has been used for the nanolipid carriers, and this is a piece of work from the lab which is being funded by ICMR, and this is the drug candidate Tacrolimus, is a novel immunosuppressant, and the challenges are as we have mentioned about, and the strategy is the NLC. The screening of liquid lipids was done, and the screening in which we generally find out long chain oils and the medium chain oils, because they are being reported that they have a good lymphatic uptake, and accordingly we try to have all those which are available for that purpose only. So it exhibited a poor solubility in long chain triglyceride, right? And it was having a good solubility in the medium chain triglycerides as such, right? This is screening of solid lipid, where we just try to find out what is the amount of drug which can go into the solid lipid, and accordingly, one of the lipid was selected as a solid lipid which was having the highest solubility of the drug in itself, right? Then we did the screening of surfactant by aqueous titration in order to find out from the ternary phase diagram between different formula components of nanoemulsions, right? Lipids, water, and surfactant, and they were used in various ratios, right? Alone also, to find out what is the maximum area of emulsification which we can achieve, right? So the yellow dots are representing the crude emulsion area, and the different areas of nanoemulsions were obtained using different surfactant or surfactant combination. And accordingly, we came to the conclusion of using Campule MCM-C8 and Computerol 888A-TO into 2:3 ratios of lipid phase. Surfactant was a combination of solutol HS15 and lecithin. The concentration range of lipid was 1 to 2% weight by weight, and concentration range of surfactant was 1 to 3% of weight by weight. Since tacrolimus also have the problem of degradation if it is exposed to a high heat, so we use a conventional solvent emulsification evaporation method and a conventional high pressure homogenization method, that is method A and C. And then we did a slight modification in order to decrease the heat generation by modifying method A as method B, and method C modification leading to method D. In this, in the earlier two method, the probe sonication time was decreased and the amplitude was also decreased. In the method C, in the method D, we used less number of cycles so that at a less reduced pressure, so that heat generation is less. So conventional method, lipid mix phase was taken, right, and the aqueous phase was taken. Pre-emulsions, right, mixture was passed through HPH or probe sonication, right. So this is the conventional method, right. In the modified method, what we did was pre-emulsion was taken and cold aqueous phase was added. Half was one done. So first hot aqueous phase in the half solution was added, and later after formation of pre-emulsion, a cold aqueous phase was added, and then it was passed through HPH or probe sonication. So that was a major modification we did. And then we just try to find out the composition of NLC formulations, right? And then the we in series of formulations were created, and they were seen for optical clarity, mean viscosity, and then percentage transmittance, which were giving the highest value. We just tried to pick them up. And the mean particle size, our aim is to go till 200 or maybe plus minus 20, right? So those series of formulations were selected. They were having mean PDI. PDI, I think for your information, it is a polydispersity index. And this talks about homogeneity within the particle size range. And the value should be less than 0.5 for an ideal one, but it can go up to 1 also. But we have selected till 0.5. Percentage entrapment efficiency was talking about the drug which you used for the theoretical loading. It was calculated that how much drug has been taken from the has encapsulated in itself in the nanolipid carrier. The loading efficiency talks about the total output theoretical content, which is generally reported not more than 10%. Accordingly, it was also satisfactory. Out of these formulations which were taken, right, we used preparation method B and D, and the mean particle size which were selected was like 70 and 98, and then PDI value less than 0.5, and accordingly we have a good entrapment efficiency and loading efficiency as well. The modification in the solvent emulsification evaporation probe sonication method resulted in much reduced sonication time of 2 minutes at an amplitude of 20%. And in the case of modification of HPH, we were able to achieve a good particle size and the PDI, which was of importance, with 3 HPH cycles and 300 bars pressures. Now, we took these formulations further for characterization. The characterization parameters are based on the surface topography and then interaction studies to study about. So SEM and TEM results confirm that these are prepared by modified method were found to have shapes and smooth textures, right? And DSC basically inferred us that it is demonstrating encapsulation of drug within NLC in addition to the solid state of the NLC as such. FTI results confirmed that structural integrity of the drug was also intact. After this, we entered into the in vitro release pattern, that is the release being studied at the simulated gastric fluid and the simulated intestinal fluid, SGF and SIF, without pepsin and pancreatine, right? So these are simulated one. And these were tried. The drug was incorporation in the imperfections of binary lipid structure resulted in greater amounts, more than 95% of released drug as compared to drug suspension 10% and marketed formulation 16% up till 24 hours. So we were able to achieve a sustainment of action, which was good, and then the percentage of the drug release was also high. In vitro lipolysis studies that talked about where we used a degradation media in order to undergo a lipolysis process, the higher amount of solubilized drug from the nano formulation in the aqueous phase indicated that much drug, which is more than 60%, would be available in solubilized form for absorption after oral administration. So that was also an indication that we'll going to get a good in vivo prospects as such. Coming to the in vivo studies in terms of the bioavailability studies and the lymphatic distribution studies, these studies reveal the tacrolimus was estimated in rat, whole blood, lymph node, and other organ homogenates. through an
आया थोड़ा सा
HPLC MS chromatography system, and we just try to calculate the levels of the drug. The increment in the relative bioavailability of the nanocarriers was found to be 6.2 and 2 folds with two different formulation, which was outcome of the propsonication and outcome of the HPH formulation, right? And this was better than that of the drug suspension and the marketed formulation respectively. And these were the pharmacokinetic parameters which were obtained. And here, if you see the AUC levels, there was an enhancement of the levels 6 times and then 3 times according in the two optimized formulation. And the other parameters were also improved in terms of pharmacokinetic as such. Then the correlation of the in vitro lipolysis and the oral bioavailability results were also done in order to find out whether the in lipolysis oral bioavailability, has any connection. And it was found that the higher the level of the amount of drug available for absorption, then you get a good correlation also. So that was also being reported. And this is the in all organ distribution pattern, organ and leaf distribution studies, and these are the in heart, lung, liver, spleen, kidney, stomach, intestine, and lymph as well. And this result demonstrated that lymphatic distribution of NLC was significantly enhanced by 19 and 14 folds at 1 hour, 7.5 hour, and 6.8 folds post 24 hours respectively, in contrast to the suspension. So it was better taken in the lymph node lymph system, which was also connected for the systemic circulation as such. So this is a brief schematic representation that how both the NLC formulations, talked about the cumulative drug release, right? This is a drug suspension, which we are talking about. And what is a pattern in the case of release in pH 7.4? This is the simulated SIF, right? And this is with the drug. And then talking about the amount in the blood and the lymph and the liver, and accordingly in other major organs as such. And this was an overview, right? And in conclusion, the concentration of tacrolimus in blood and lymph were significantly improved, attributed to the medium chain triglyceride, the long chain triglyceride, because we saw that the solubility was higher in case of medium chain triglyceride, which have also been reported for a good lymphatic uptake. The study demonstrated that NLC of tacrolimus is an extremely potential approach for enhancing its therapeutic prospect and should be explored commercially also. Now, this is one of the publication out of this work where we publish an overview of certain immunosuppressive drug therapy, and NLC as to overcome the challenge and remedies was talked about under this expert opinion. And this is the work, research work, which was published in European Journal of Pharmaceutics and Biopharmaceutics on in vitro optimization and characterization. And this is talking about the in vivo bioavailability enhancement in terms of all routes of the body and body distribution, organ distribution, and so on. And in conclusion, NLC is a potential approach for improving the oral bioavailability of highly lipophilic drug with poor aqueous solubility, extensive first-pass metabolism, affinity of P-glycoprotein efflux transporters, and susceptibility to intra and prosite metabolism. And they have a high caliber of showing, better drug loading and stability. Disrupted matrices of NLC, that is imperfection, they are leading to what we have talked about, a higher drug entrapment, a good modulation of drug release, and enhanced drug absorption. These special features of NLCs are exclusively attributed to their unique composition, which is constituted of a blend of incompatible solid and the liquid lipids as such. Future prospects, right? The promising results obtained in animal models suggest that the formulation should be extensively tested in human subjects. Still, some formulations are under trials. In vitro model studies, preclinical and clinical data on a wider range of drug candidates with different physicochemical properties would help the establishing the mechanism or elucidating the mechanism of action of the NLC formulation. Exploration in improving the absorption of highly lipophilic drugs in conjunction with thorough understanding the effect of formulation components on the efflux transporter, metabolic enzymes, and lymphatic uptake would help in completely addressing all the negative aspects of oral delivery of lipophilic drugs. And with this, I end up with my presentation. And if you have any issues or any clarification needed, I'll be here to help guide.
Thank you so much, sir. Sir, we have few questions. So one question is from Ira Malia Khan.
Yeah.
She is asking, can you enlighten about stability study of nanoformulation?
Stability study of nanoformulation, yeah. So I think, I have already covered up in terms of physical stability. It has the highest stability in the series of all lipidic, type of formulation. And the reason because, these structures so formed, imperfectional drugs are keeping themselves. So they have a high physical stability. However, in order to establish the chemical stability and avoiding its shelf life, you have to use a protocol, maybe an ICH guideline or a US FDA protocol in order to assign a shelf life according to the drug characteristics, that what is the nature of the drug and how the stability differs for that drug. That also needs to be established, as we are doing for other formulations also. It's like that, right? So physically, it has a greater stability, right, when you compare it with other colloidal vesicular drug delivery system. But in order to establish the chemical stability, you have to use the according protocol of the regulatory body. Thank you.
Thank you. So one more question. Sir, a method to find out recrystallization index other than XRD and DSC, which we can perform in college laboratory.
So see,
This
crystallization
is
index.
from Alok Pratap Singh.
Okay, so crystallization index, it will be difficult to find out without the help of the instrument, because you cannot quantify with the eye, right? Maybe you can do it visually, but it will not be acceptable when you are communicating your manuscript. I think it is to be backed up via instrumentation data. Maybe XRD and the DSC is more being utilized, and you will find many papers on crystallization index reported from the point of view of the DSC data or XRD data. So in lab, maybe you can do a hit and trial just to quantify in terms of your visual outcome, but difficult to be quantified without an instrumentation, right?
Okay, sir. So that's it. Thank you, sir, for giving insight on this burning topic. Nanotechnology has expanded exponentially in every field. Nanostructured lipid carriers have attracted expanding scientific and commercial vigilance in the last couple of years as alternate carrier for the pharmaceutical consignment. It is foreseen that in near future, nanostructured lipid carriers will be further advanced in a more efficient, exact, and protected manner. Our participants will be definitely benefited with the extensive research experience that you have shared. Thank you once again, sir.
Thank you for the opportunity, and I trust and I have a confidence that some of the points, I understand that if you're hearing a topic first time, so that is not going at a spontaneously. That's need perseverance, right? And then you are what I should say, impressed by the way you can prepare. There's a lot of literature available, and according to the literature availability, you can just try to explore, and you can also have good publication. You can have good project funding also from this area. After this ICMR immunosuppressant project sanction, right, we also had a grant where we have also investigated few of the antiretroviral drugs, particularly which are being highest generation, second generation drug candidates for the treatment of HIV and so on, right? So all the best, and thank you for your patience listening. Look forward meeting you all sometimes again. Thank you.
Thank you so much, sir.
Thank you very much,
Thank you, Praveen ji. Thank you, Praveen ji.
Thank you, sir. Thank you, sir. Thank you, sir. Thank you, sir.